present regulation of stomata. Very, very interesting research.
Okay. In the next 10 minutes or so, I'm going to present an example of how the lysimeter scale system helped us resolve and overcome a major bottleneck that we had in our study while trying to evaluate plant performance under drought. Today my lab focuses on citrus molecular physiology and genome editing, but I want to take you back to my PhD where I studied sugar metabolism and stomatal movement. So I'm going to start with why studying the link between sugar metabolism and water transport, why we think it's important, and what we have achieved in this study. And then I'm going to present two major steps in our project that improve the precision, the accuracy of our study. One is using tissue-specific promoters, and the other one is using the lysimeter scale system to resolve transpiration inconsistencies that we had while using instantaneous measurements. So plants are facing an ever ongoing conflict between the need to open the stomata to allow CO2 intake for photosynthesis and sugar production, and the requirement to close the stomata to prevent water loss. So whenever stomata are more closed, plants obviously lose the ability to perform photosynthesis. So in the beginning of our study, it was known that high levels of sugars can feed back to prevent or to limit photosynthesis. And it was known that the hexokinase, here there in orange, the hexokinase serves as a sugar sensor within the plant that senses high sugar levels and responds with a feedback inhibition on photosynthesis. And we asked a simple question in the beginning of the PhD, whether the same sugar sensor or the same high sugar levels can have similar feedback inhibition on transpiration. So we asked two main questions in the beginning of the study. One is whether sucrose, the main transported sugar in most crops, can limit stomatal movement. And second, whether hexokinase itself, the sugar sensor, can do similar things. So in the beginning of our study, we saw immediately that sugars closed stomata. This changed the paradigm. Up until then, it was thought that sugars actually open stomata. We proved the other way around. What we saw is that sucrose itself can generate an ABS signaling within the stomata, leading to stomatal closure. And in addition, we saw that it dependent on the sugar sensor, hexokinase. So overall, sucrose induces stomatal closure, and it does so via hexokinase and ABA. So then we said, okay, we have a pointer here. Okay. Okay, can you see it? Then we ask, well, if sucrose can induce stomatal closure, how about the enzyme itself? So in that case, we use transgenic plants that was generated by Nir Dai way back. And we saw indeed that in correlation to the elevated expression of hexokinase, this sugar sensor, we can see the stomata are more closed, and we can see that stomatal conductance is reduced. And then we ask, okay, this is nice. We can obviously see an effect of hexokinase on transpiration, but what about the urinary performance? How those plants act and behave throughout the day? And here I will take you back to the initial prototype of the lysimeter. This is the original system. This is how it all started, and I was one of the first users of the original system. It was like a table with 6 panels, and that's it. And this is how we measure the transpiration, and everything was manually. And as you can see, the correlation, nice correlation between hexokinase expression and transpiration rate. So we have now a nice way to save great amount of water while overexpressing the exokinase. But we have one major issue in these plants, that in these plants growth was severely impaired. So we can save water on one hand, but we have growth impairment on the other hand. So what we did, was one of the major jumps or major leaps that we did in the studies. Instead of expressing exokinase throughout the plant, we express it specifically in the guard cells, assuming that if exokinase indeed senses high sugar levels and responds with stomatal closure, let's remove all the negative influence that exokinase has in other tissues. And this is exactly what we did. We generated tomato and Arabidopsis plants, and you can see here in green, GFP serves as a marker. You can see that the promoter was highly specific to guard cells. And then we started with instantaneous measurements while working with the LICO, and you can see that plants that expresses exokinase specifically in the guard cells, you can see that stomatal conduction and transpiration were indeed reduced, while photosynthesis remained unaffected, thereby improving the overall water use efficiency, which is basically the balance between carbon gain and water loss within the plant. So this was very nice, and we had a nice bonus, as we thought, compared to 35S promoter and expressing throughout the plant, which had severe growth inhibition. These plants actually had no penalty in growth, and sometimes the growth was even enhanced. So that was all very nice. But then we ask a simple question: what about drought? If we want to offer this technology in any way, we have to study it under more field conditions, and we experiment the drought. And when we started our experiments, using LICO or using porometers, it was a huge frustration there. In some of the experiments, the stomata of the transgenic plants were more open. In other experiments, the stomata were more closed. So we couldn't draw any conclusion or any good conclusion about these experiments, and it took us a lot of time. We were struggling with it until we went for the lysimeter scale again, and this time we performed high throughput in the more advanced system. And I'm not going to give you all the parameters that the lysimeter scale can measure. I'm just going to give you the bottom line. And this is actually the results that now we understood why in some of the experiments stomata were more closed and some of them they were more open. So as you can see, it's divided into two stages. One is the normal stage. Let's call it mild drought. You can see the stomata of the GC exokinase plants are more closed. That leads to water savings and high water use efficiency. And at the second stage, the stomata of the transgenic are more open compared to the wild type, which responds to more severe drought with a steep decline in transpiration. So overall, we think that the gap between 1 and 2 provides some advantages for these plants if we're talking about mild cycles of drought. So that was a nice understanding for us to know why in some of the instantaneous measurements we couldn't tell whether they are open or closed. In Arabidopsis we saw similar behavior, similar pattern. Again, overexpression of the exokinase specifically in the guard cells. You can see that stomatal conductance and transpiration are significantly reduced. Photosynthesis remained unaffected, and water use efficiency rises together with better biomass accumulation. And then we went to another high throughput experiment, and I think we were one of the first group who tried Arabidopsis on the lysimeter scale system. And this is David Granot, who was the PI of the study, together with Menachem. And then we did this experiment for the first time. We didn't have high expectation from Arabidopsis. It was tricky, challenging, but then after you set up the experiments in an elegant way, Then the experiment runs, and there were no issues. Everything was smooth, and we were very happy with the results. And we have results in this experiment only for the control conditions. But again, you can see that throughout the day, the transgenic plants display lower transpiration rates in 3 consecutive days presented here. And so overall, with regard to drought, what we think that happens in the transgenic plants is that the transgenic plants simply delay the entering into the drought phase. You can see that the wild-type plants in general, at the first stage, the stomata of the transgenic plants are more closed. This is in mild drought or early stages of drought, and we can save water and have higher water use efficiency. But later on, we see a flipped pattern. The stomata of the transgenic plants are more open and simply displaying delayed exposure to drought while maintaining high productivity. So again, if we have these cycles of mild drought, we think it will provide a nice advantage while using these plants. So once we did the experiments on the lysimeter system, we never stopped using the lysimeter system because it was just so convenient. You just put the plants and then you forget about them and you get the results after 2 or 3 weeks. And we did it in citrus and tobacco. This is the work of Nitzan Lugassi in our lab. And I did it also in barley. You can see in tobacco, again, you can see the flip between the wild type, between the normal and the drought, that the wild type just steeply drops while the transgenic plants maintain, let's call it drought avoidance. So in general, from the scientific point of view, when we overexpress the sugar sensor, we can save water, but we have growth inhibition. But when we express it specifically in the gout cells, we can save water, we can enhance water use efficiency, and we can have drought tolerance. So overall, we saw in our study that sucrose and the sugar sensors can induce stomatal closure. We suggested that exokinase serves as a sensor that coordinates between photosynthesis and transpiration, and that if we can use this technology, we can have better performance with regard to transpiration, water use efficiency, under normal as well as under drought conditions. And on our hand, the lysimeter in our study made all the difference. We had a lot of struggles before, and then when you see the diurnal behavior and long-term behavior, it made all the difference for us. It made my life way more easier. So just final thank to the team of David Granot back then, Menachem Ushalion back then was still in touch, of course, and of course to Rakefet, and to the super technician of our lab, Ayman Egbaria, which took the heavy load of all these experiments that I just presented. And thank you.